Alteration or changes in the structure of individual chromosome is nothing but chromosomal aberration. This may occur spontaneously or by induction. Such changes may result in quantitative alteration of gene or rearrangement of genes. The breakage and reunion of chromatid segment results in the number of abnormalities in the chromosome structure. Thus, origin of structural changes is caused by breaks in the chromosome. Let us go to the types of chromosomal aberration. There are four different kinds of structural changes of the chromosome have been demonstrated. Number one, deletion. Second one, duplication. Third one, inversion. Fourth one, translocation. Number one, deletion. This includes part of chromosome will be lost or deleted. It causes changes in the number of genes. Second one, duplication. It includes part of chromosome added or duplicated. This may also causes changes in the number of genes on the chromosome. Next comes inversion. Here, section of chromosome will be detached or reunited in reverse order. It causes changes in the arrangement of gene on the chromosome. Last one, translocation. Here, part of chromosome will be detached and joined to non-homologous chromosome. These changes will affect the structure of chromosome and cause problem in growth, development and function of body's systems. These changes can affect many genes along the chromosome and disrupt the proteins made from those genes. This picture tells about all types of chromosomal aberrations such as duplication, deletion, inversion and translocation. So in this picture at the top one can see normal chromosome having 6 genes designated as A, B, C, D, E and F. In duplication one can see gene A is repeated once again to the adjacent of another gene called A. So here in case of duplication, a gene is added, extra gene is added to the normal chromosome, that is nothing but A. So you can see 7 genes on a chromosome. This is called as duplication. If you go to the deletion, there is a loss of a gene called F. In the picture clearly you can identify there is a loss of a gene called F when we compare to the normal chromosome. This is called as deletion. It is a type of a chromosomal aberration. If you go to the inversion, in the picture one can observe very clearly there are some genes called DEF are inverted when we compare to the normal chromosome. So this is called as inversion. So in translocation, in the picture, one can see a extra segment called XYZ. So this shows there are some segments which are transferred from some other part of the chromosome to the normal chromosome. So this is a type of a chromosomal abbreviation. Let me see deletion in detail. Deletion represents a loss in chromosome material and was first chromosomal aberration indicated by genetic evidence. This evidence is presented by Bridges in 1971 in Drosophila melanogaster and showed a deletion of the X chromosome that include the bar locus. There are two types of deletion. Number one, terminal deletion. Second one, intercalary deletion. These two types of deletion depends on number of breaks commonly seen on the chromosome and then responsible for deletion of particular genes. Example for deletion is Cry-do-chat syndrome, where part of the number fifth chromosome is missing or deleted. Babies with this syndrome shows high-pitched cry, poor muscle tone, a small head size, and a low birth weight and problem with language. Along with this, delays in walking, problem with feeding, hyperactivity, and severe intellectual disability is seen in cry-do-chat syndrome. The term duplication simply means that a part of the chromosome is duplicated or present in two copies. This results in having extra genetic material even though the total number of chromosomes is usually normal. These extra copies can lead to errors in the development of the baby. One example of a rare genetic disorder of duplication is Pallister-Killian syndrome, where part of the number 12th chromosome is duplicated. This picture tells about the difference between deletion and duplication. In deletion, there will be the deficiency of a gene, whereas in duplication, there will be addition of a particular gene. Duplication are of different types on the basis of position of duplicated segment. There are five types of duplication. Number one, tandem duplication, displaced homobranchial duplication, displaced heterobranchial duplication, transposed duplication, at last reverse tandem duplication. This picture tells about different types of duplication. Okay, if you carefully observe, you can see the lot of difference between all the five types of duplication. Type of a syndrome due to duplication of a particular gene on chromosome number 12 in human. You can see there is a lot of difference in the facial expression, in the lips, in the forehead. So this is about the duplication. Let me go to another type of chromosomal abbreviation, inversion. Inversion represents reverse gene order in the chromosome. Inversion originate when parts of the chromosome become detached and turn through 180 degree and are reinserted in such a way that the genes are in reversed order. There are two types of inversion. Number one, pericentric inversion. Second one, paracentric inversion. In pericentric inversion, this inversion occur when the inverted segment that includes this centromere. Paracentric inversion occur when the inverted segment does not include the centromere. One of the best characterized recurrent inversions giving rise to disease causes hemophilia A, an X-linked disorder caused by the mutation in the factor 8 gene. In some cases, it has been associated with congenital anomalies, growth retardation, infertility, and recurrent pregnancy loss and cancer. Number chromosome 9 inversion, also known as inversion 9, is related to Walker-Warburg syndrome and acute leukemia. This picture clears the paracentric inversion and pericentric inversion. This picture shows a baby suffering from Walker-Warburg syndrome due to the inversion 9. Let me pass on to last chromosomal aberration translocation. Sometimes a part of a chromosome becomes detached and joins to the part of a non-homologous chromosome, thus causing translocation. A translocation occurs when a piece of one chromosome breaks off and attaches to the another chromosome. This causes chromosomal abnormality whereby there is a break in the chromosome, one particular chromosome, and that chromosome will then fuse to the different chromosome. So this type of translocation is responsible for some type of cancer, example leukemia. Let me go for types of translocation. There are three types of translocation. Number one, simple translocation, shift location, and last one, reciprocal translocation. So this picture shows about the translocation in heterozygote as well as in homozygote condition. Simultaneously, this picture tells about the translocation of some genes between two homologous chromosomes. Overall, one of the most frequent cause of evolution is the change in the order of genes as a result of chromosomal aberration. Thus, chromosomal aberration play a vital role in evolution as they generate variation in a natural population.